Hey guys, so continuing to talk about lymphocytes. So last week we talked about B cells and how macrophages or dendritic cells can gobble up antigens. They put them on MHC class 2, and then they present that to a T helper cell, which has a T cell receptor that is very, very specific. There's only like 1 out of 1 million will have a T cell receptor that actually binds to MHC class 2. We're going to talk a little bit more about that today. And then that T helper cell then goes on to look for a B cell that has that exact same MHC class 2 with that peptide, okay? And so that sort of connection that happens here, it doesn't have to happen at the exact same time, though it can, but that triggering actually allows the B cell to become activated if it was initially primed using its B cell receptor. And we talked about that last time. And so you would start generating antibodies. But now what we're going to talk about today is actually how lymphocytes develop. And then we're going to come back and then we're going to talk about how T killer cells, which are down here, so these are your CD8 positive T cells as opposed to CD4 positive T helper cells. Your CD8 positive T cells are your T killer cells, and they're going to go and look for virally infected cells, or they can be cancer cells, and blow them up, a lot like how NK cells do. But before we get there, we need to talk about how these lymphocytes actually develop, and that's a really, really cool thing. So all lymphocytes develop from the hematopoietic stem cell system. I've talked about that before. I've shown a picture of just that descent from an initial stem cell to a hematopoietic stem cell, and then you have your myeloid or your lymphoid progenitors, and then they can become all those different cells. We talked about that in the innate immune system. Your lymphocytes come from the lymphocyte lineage, but they have to be trained first. So the way to think about your lymphocytes is that they are incredibly specialized and powerful weapons that have to be used sparingly, because to actually use them requires a ton of energy, and they can be incredibly powerful and dangerous if you accidentally let them loose on an antigen that maybe is a self-antigen. So for instance, let's say you accidentally trigger a B cell response and make antibodies against your myelin sheath cells. Well, now you have just killed all of your neurons and they're all going to die, and now you're toast. Okay, so that would be a really bad thing, right? And so we need to talk about how we actually train our B cells and our T cells, which the T cells is actually better understood how we do the training, and how we train them to avoid attacking you. So all of our lymphocytes start out in the bone marrow. So you can see you got a nice little femur here. It doesn't have to be in the femur, but anywhere. And the B cells are going to hang out there. So they develop, they become pre-B cells. They are going to be B cells, but they just have a bunch of different processes to go through to actually develop. And so they're going to hang out in the bone marrow and do their training. But we need to follow T cells. So the T stands for thymus, and T cells are going to traffic to the thymus. So you can see on this page here, they go through the blood, head to the thymus, and they're going to get trained there. T cell training occurs in the thymus, as I keep saying. So the thymus is this weird looking organ. So there's your heart and there's your lungs. Okay, and you've got your heart here, and the thymus sits on top of the heart, on top of the pericardial sac. There's a lot of fat that actually exists up there. They're not really showing it justice. I like this picture a little bit better because it's really showing what the thymus looks like, at least in a young person. So the younger you are, the larger your thymus is, because you need to be ready to train tons and tons of T cells to be able to fight off infection. As you get older, your body's already kind of used to fighting off a lot of viruses and things, and so you don't need as big a thymus because, I don't know, why do you need to train your T cells if you've already, fought off 80% of what you're having to fight? And so that's why, like in old people, you can't even find a thymus. It gets hard to find because there's like a fat glob there, and the thymus, it really does have this color. We used to pull the thymus out of mice, and it was really hard to find. So you would just take that whole fat glob that's sitting on top of their heart, and then you would put that, you actually can put that in like PBS, which is like a saline solution, and lymphoid organs will sink because they're really dense, and fat will float. And so that was one way to separate it. So there's your fun fact of the day. Now, your thymus, it sits on top of the heart, and it's going to do our T cell training. Well, what does that really mean? Well, we need to talk about what that means. And to talk about that, we need to understand what makes a T cell look like a T cell. And I've talked a lot about, let me go back to this picture, our T cell receptor. Okay, your CD4s and your CD8s, they both have T cell receptors, and they're incredibly specific to the antigen that we're trying to see. And so in this case, we need to talk about the difference between a T cell receptor, okay, and a B cell receptor, which is something else that I talk about a lot. So last time we talked about B cell receptors, they're just antibodies on the surface, right? So let's look at them together. So on the left, we have our typical antibody. This one is membrane bound, so we call it a B cell receptor, but it's just an antibody. And the portion that we're really concerned about is this portion here. So you'll notice it's got these two chunks right here, okay? So this is your variable region and your constant region. They're implying that this is constant. I mean, I guess you could say that. So let's just say this is our variable region, this entire thing up here, and then you've got your constant region here, and then we're going to replicate that in our T cell receptor. So these are almost identical in how they are generated. The exact same set of genes is going to code for this piece of protein. Now, yes, B cell receptors or antibodies, they have two binding portions that are identical. That's just so that we can grab onto multiple things. But T cells aren't secreting their antibodies, so why have two of these? We're just going to have one. And so you're just going to basically take this portion of the antibody and then kind of take it by itself and stick it onto the surface of a T cell, and that becomes our T cell receptor. It's still incredibly specific for an antigen. Just to show you another picture of what the structure looks like, a little bit of a better picture. So this would be where you would actually bind the antigen, and there is a constant region. So that's why I got a little confused here. They're saying that this is just the constant region. I mean, you could actually argue that this bottom portion here is the constant region, but that's fine. And so here's our T cell receptor. There is an alpha and a beta side. They're two separate chains that have to come together. They dimerize. And then that's what your T cell actually puts on the surface, and it goes around looking for MHC. If it's a T helper cell, it looks for MHC class 2. Right? That's what we've been talking about, right? T cell receptors have MHC class 2. So here's our T helper cell. It's looking with its T cell receptor for MHC class 2 with an antigen that it recognizes. T killer cells, almost the exact same. They're just looking for antigen in the presence of MHC class 1. And their actual structures provide them that ability to decide, okay, do I bind to MHC class 1 because I'm a T killer cell, or do I bind to MHC class 2 because I'm a T helper cell? Now here's where we actually need to talk about the training of T cells, because it's really important. Keep in mind, the main point of all of this is that we need to make sure that we don't have self-reactive T cells or B cells. Okay, now we're focusing on T cells here. So we can't have self-reactive. What does that mean? If you have a self-reactive T cell, then it might accidentally induce an autoimmune response, and that would be really bad. So we need to talk about how we actually train our T cells, and we're going to use our T cell receptors to kind of teach us how to do that. And so let's look at this diagram here. On the top is positive selection, and on the bottom is negative selection. Those are the two levels of training that we have our T cells go through. This is occurring inside the thymus. And so let's back up real fast and show you this diagram again. And the bone marrow is where we make our T cells. They head through the blood. They're heading to the thymus where they need to get trained, okay? And so in the thymus, they're going to undergo two levels of training, positive and negative selection. And these levels of selection actually make a lot of sense. Can your T cell receptor actually bind to MHC, either class 1 or class 2? We test both. Why is that important? Well, because you don't want a T cell receptor that doesn't work, because what's the point? You're just wasting resources building these T cells that will never be able to trigger an immune response. So we know we have our T cells and we know that they work properly, at least that their T cell receptors will bind to MHC. Okay, awesome. So they can receive a positive signal. But the problem is, what if they receive that signal and they're binding to MHC? That's fine. But what if that is self-antigen? So remember that an antigen is typically a protein that can be basically anything, and it's going to bind to a specific chunk of that antigen, or what we call an epitope of an antigen. And everything in your body can basically be an antigen. And so self-antigen would be like an autoimmune response. So if I have a T helper cell that can bind onto a self-antigen, or a T killer cell that can bind to a self-antigen, that would be really, really bad. Because now I'm able to trigger an immune response against a self-antigen and trigger an autoimmune disease. So that would be a really bad thing, right? And so that's the negative selection process. So I take my T cell, I show it on its T cell receptor a self-antigen. I have another diagram here just to kind of show you what's actually happening inside the thymus. And so on the right here, we have our precursor. So this would be your pre-T cell. It heads in here. and it's going to go through a few different steps. So DN stands for double negative because they don't actually have CD4 or CD8, because that's how we distinguish if it's a T killer cell or a T helper cell. Or T helper cells have CD4, T killer cells have CD8. So if you don't have either one of those, you call double negative because you don't have either one. And early on, they actually express both. Because it's actually poorly understood how we decide, does this T cell become a T helper cell, or does this T cell become a T killer cell? It's actually not really well understood. There's a lot of theories, but we don't know. But we do know that early on, they express both. And so they make sure that at least we're going to go through positive selection. We take their T cell receptor, and a special epithelial cell called a cortical epithelial cell is going to take an MHC class 1 or class 2, and it's going to just make sure that that T cell can bind onto it with its T cell receptor. Because if it doesn't, well, then what's the point? And so if it doesn't bind, okay, because that would be bad, because that means it can never receive a positive signal. So if it doesn't bind, it dies by what we call death by neglect, because it's not receiving a positive signal. And so if they just kind of sit around long enough, they eventually die because they never receive a pro-growth signal. But let's assume that it did work. So their T cell receptor works. So we had a positive selection. And so we decide at some point we commit to either a CD8 or a CD4. It's poorly understood why that happens, but we switch from a double positive to a single positive. Okay, so either CD8 or CD4, and it's saying committed. So sometimes they can still have the other receptor on their surface, so it could be CD4 positive and CD8 positive, but we know that it will become a CD4 positive or whatever. That's why it's saying it's DP for double positive, but hey, what you gonna do? So we take our CD8s and we send them on, and we take our CD4s and we send them on, and we're going to check and see if they can bind self-antigen. And that's where a special cell called a medullary, because it's in the medulla of the thymus. We're not going to go through thymic structure in this class, but it just has a portion that's called the medulla, or the medulla if you want. And they have medullary epithelial cells or medullary dendritic cells, and they have such a cool job. Why is it so cool? Because I didn't learn this until later, and it actually informed a lot of my understanding about how this actually works. So we know positive selection, when we go through positive selection, we actually are, making sure the T cell receptor works. But when we go through negative selection, how in the world are we going to take self-antigen, right? So those are proteins that we're supposed to take from the rest of the body because they're self-antigens, right? You're the host, and we're supposed to take these antigens and chop them up and present them to these T cells. How in the world does that happen? And it happens through one of the coolest mechanisms that I know of. So we're going to zoom in on what one of these cells actually looks like. So they're calling it a medullary epithelial cell, or it can be a medullary dendritic cell. And you can see here, we've got our tissue-specific gene, and what's going to happen is this AIR gene, is what it's called, is going to come into the nucleus. Okay, so it heads into the nucleus, and it's going to find a random protein to generate. Okay, so here's our coding region for that specific gene, and it's going to use its special abilities to promote the transcription of this. How it actually does this is super duper interesting. And so over here, it's actually talking in a little bit more detail. This is still poorly understood how exactly this happens, but you have this AIR protein come in here, and it's going to bind to a region. It's often a region that is condensed. So if you take a genetics course or biochemistry course, we talk about the density of chromatin. Okay, because chromatin is histones and DNA, and if it's really dense, it's hard to access, and if it's really loose, it's really easy to access. And the AIR gene actually heads towards genes that are hard to access. So it has to open up the actual chromosome structure and then trigger transcription, which actually makes sense because in this medullary epithelial cell, it's got all of its proteins that it's normally expressing, and it can show those to T cells. But if it wants to find something weird, like let's say like a neuronal protein, or I don't know, some other cell type's protein that's not being expressed normally in a medullary epithelial cell, well, it's going to go and look in its chromosomes where the chromosome is really condensed down, because we know that's a way to sort of control what proteins are expressed in a cell. If I take my chromatin and I condense it down really tight, then my RNA polymerases aren't able to get in there and transcribe that, and therefore I won't express that protein. So if that was like a neuronal protein, then I'm not going to express it, and therefore, that makes perfect sense because this isn't a neuron. Why would it express that? And so the AIR gene actually goes for those areas to look for things. And so it's promoting the exposure of T cells to antigens that are normally turned off in a medullary epithelial cell, but are normally turned on in other cell types. So jumping off of that, I want to back up just a little bit again and go back here to this structure. Okay, actually, let's back up here. So we've got our B cell receptor or our antibodies. We've got our T cell receptor here. Now we know how important T cell receptors actually are because they're the business end of a T cell, right? They're what's going to grab onto an antigen in the presence of MHC, and it's going to trigger a potential immune response. If it's a T helper cell, that's awesome because that can then go on and tell a B cell to do its job. If it's a T killer cell, then it can receive a positive signal from a dendritic cell and then go on to look for that exact same antigen with MHC class 1 on a virally infected cell or a cancer infected cell, if you will, and destroy it. So we need to talk about how T cell receptors and B cell receptors are actually made. And that kind of goes off of this. So you can see we've got our variable region, we've got our constant region. There's subdivisions of these actual proteins that are coded by genes. And there's also alpha and beta chains. So let's take a look at how these are actually made. Now on this next slide, there's a lot going on, but I'm going to take you through it because it's actually really, really cool. So on the left side here, let's just focus on the antibody here. So on the left here, we have our light chain. So the light chain is the smaller chain. It's literally smaller, so it's literally lighter. And then we have our heavy chain over here, which is a lot longer, thus heavier. And we have our antibody, this is the binding portion or the variable region, and then you have your constant region. And so we're going to focus on the variable region. The variable region needs to have a lot of diversity. So it needs to be able to bind literally any antigen on the planet. Next slide has a lot going on, but I'm going to take you through it because it's really, really cool. So on the left side here, we're going to focus on our antibody, and we have our heavy chain, which is longer, thus it's literally heavier, and then we have our light chain right here. And this is the business end of our antibody, right? That's the end that's going to actually grab onto an antigen or an epitope on an antigen. Now that needs to have incredible variability because we want to be able to potentially bind any possible antigen in the known universe. And so we're going to have a ton of variety here, or potential variety. And so what we're going to do is we're going to pick one variable region gene, is what it's called, and we're going to pick one J region gene, which is called joining, because it joins the constant region to the variable region here. Okay, so you can see it says constant here. And so what we're going to do is we're going to pick one of 36 potential variable regions, and we're going to pick one of 5 potential joining regions to stick together. And so right off the bat there, you have a ton of diversity potential. So all of these genes actually have different amino acids that they code for. And so you can pick a ton of variety here to actually generate just one side of this to have a ton of different specificity for different antigens. The same holds true as well for the heavy chain. So we got our light chain there, so we got our heavy chain, and the variable region of our heavy chain has a variable set of segments. So we've got 39 of those potential ones. There's the joining one again, so there's the joining. And then in between, there's even more diversity, and that's why we call it the diversity segment. And you're just picking one of these genes. So there's 27 in a row, all with different structure. And you're going to pick one of those and stick it in between one of these variables and one of these joining segments. And then stick that onto a constant region, which there's some variety there, which we're going to talk about in a little bit. And then that's what gives you so much variety in this actual binding segment here. That's really, really cool because antibodies are generated this way, but your T cell receptors, which are basically just this portion right here, are made the exact same way. Now, there's the alpha chain and the beta chain, okay, because I talked about that earlier, right? There's an alpha and a beta next to each other that are sort of like dimers. Okay, so we've got our alpha on the left and we got our beta on the right. And so when we actually generate those, it's almost the exact same way. We pick one of the V segments, we pick one of the D segments, we pick one of the J segments, and then there's a constant region as well. Now, I am drastically simplifying what's happening. It's still really complex, right? But I'm actually simplifying this a lot. There's so much variety that actually goes into this. I mean, there's alternative forms instead of alpha and beta. There's tons of we're going to talk a little bit more about the constant regions and how you pick which constant region. But suffice to say, this concept should be pretty straightforward. You're just picking one random gene from all of these, sticking them together, and then that's what becomes your variable region. Now that constant region we actually need to talk about a little bit, especially in terms of antibodies. So something we talked about last week when we talked about class switching of antibodies. right? So we talked about the different types of antibodies that we can get. So they're on the bottom right here. And we need to talk about actually how do we pick which one to make. And so early on, we actually choose to make IgM or IgD. Those are the antibodies that we actually put on the surface. And so when you actually make your B cell, you start out, you pick your B cells are going to have a VDJ recombination. Okay, so that's right here. So your VDJ that you're picking for your variable region is already selected for. You can do some adjusting later on, but we're not going to really talk about that. But you have picked that portion, and so we need to decide which portion I want right here. And so I can pick my constant region to start out with IgM. So the first gene that you actually encounter is the M gene or the mu gene. It's usually written as a Greek letter. I'm just going to ignore the Greek letters and just call them by their names. And so you have your M constant region. And so that's what triggers the generation of M antibodies. You also have D there because early on your B cell receptors can be IgD, but we actually don't see that secreted too often. It's not that important of an antibody. It's just kind of a placeholder until we can actually pick a better constant region. And I talked a little bit about how your VDJ can be somatically hypermutated. That's just affecting this portion. And then the class switching is what you're actually going to pick. And so you're going to cut out between these stars here, basically. I have another diagram in a second that actually shows you what it looks like. But you're going to cut out the section that you don't want anymore. And so, for instance, if you need to make an IgG1 antibody class, well, then you cut out this M, this D, this G3, and you're left with the next gene that you're going to actually code is G1. And so you cut out this piece right here. Let's say you're trying to fight a helminth infection, like a big giant eukaryotic parasite, or you have an allergic response. Well, that means you need to make IgE. And so you would actually cut out all of these constant region genes until you hit the IgE right there. And so you can see that would be the next one that you would hit. So if I eliminate all these other ones, then you're left with your VDJ, which is the variable region, with this constant region is the next gene that you actually make. How this actually happens is a little more complicated. There's duplication units, which I am not super familiar with, but basically you can kind of ignore this part here for now. You're just going to pick, there's your VDJ segment right there. Okay, so there it is. And you're going to go along here and you can see we've got all these different terms here. So these are all just different constant regions that we can pick from. And so you've got your mu, so that's IgM. You got your gammas, that's IgG. You got your epsilons, so there's an IgE. There's your alphas, which would be IgA. So if you wanted to secrete an IgA antibody, well, then you're going to pick that segment to be the gene. And so you would cut out this other piece right here. And so that's what they're showing. It forms this circular piece, and the AID gene, well, the AID protein is actually what does the cutting here. And so you cut out this little chunk of DNA here, you get rid of it, and then you're left with a segment that only codes for the constant region that you actually want. So the next chart I want to show is just to kind of bring us back to this development, okay? So we've got our thymus, that's where our T cells undergo development. We do negative selection, we make sure that they don't bind to self-antigen. We do positive selection to make sure their T cell receptor actually works right. Those two processes, they don't necessarily occur in either order. They actually probably occur at about the same time, but it just kind of depends. It's actually very poorly understood exactly how we do all of this training, but there's some things that we do know. We do know there's a positive selection, we do know there's a negative selection. Your B cell training actually undergoes a similar process. But B cell training is actually poorly understood, especially negative selection. The idea of taking a B cell with its B cell receptor on its surface, right? It looks a lot like a T cell receptor, right? It's supposed to bind an antigen. It's not really designed to look at anything in the case of MHC. And so we would just have to somehow find random antigens and present them to the B cell. And it turns out that may kind of happen, but it's poorly understood and it probably doesn't really happen that much. I'm glossing over a lot here, obviously, because I read this whole paper and they were basically like, yeah, we don't really understand what's going on. But we do know there's a positive selection. We know that B cells, we make sure that their antibodies are constructed properly and that they can actually bind onto stuff. Whether or not that binds onto a foreign antigen is not understood, but we do make sure that they don't bind self-antigen. But this process obviously is not as easy to do with B cells, and that's why you need T cell help. That's why we have to have it where a dendritic cell tells a T helper cell, hey, look for a B cell that matches this process. Because it's harder to get rid of B cells that are self-reactive or that can cause autoimmunity. And so that's why we have T cell help. I have another chart here that just talks about B cell development. So I said they stay in the bone marrow. And as they develop, basically what we're tracking is the development of the VDJ recombination, right? So you're a pro and then a pre-B cell, and then you're an immature B cell when you actually achieved the creation of a IgM antibody. Okay, so we've talked about that as a B cell receptor, and it's undergone VDJ recombination. So we've picked that V segment, that D segment, and that J segment, and we're putting it on the surface of our cell, and it's going to head into the circulation. And so that brings us back to here. And so that B cell is circulating and looking for an antigen that it can bind onto. If it finds one that it binds onto, awesome. It's going to head to the lymph node and look for a T helper cell to grab it. Now, as I was just talking about, B cells are harder to get rid of that are self-reactive. And so a lot of self-reactive B cells actually survive that selection process here. And so if they survive that selection process, they can maybe bind a self-antigen, they can engulf it, and head to the nearest lymph node just like they've been supposed to do for all of eternity, and they head to the lymph node and they're going to look for T cell help. But we send our T cells through a very stringent selection process that we've just been talking about. And so the odds of having a B cell that is self-reactive or that can generate an autoimmune response, that can find a T helper cell that can also bind that same self-antigen is actually relatively rare. Now it can happen, of course. We've all heard of autoimmune diseases, but it's very, very rare, which is great. That's why most of us don't suffer from autoimmune diseases, because having to have this bridge between a dendritic cell with MHC class 2 and a T helper cell and a B cell is actually really, really good. So I wanted to talk a little bit about actual lymph node structure. I showed you what a germinal center looks like last time, so that's where B cells develop, but I wanted to show just a typical lymph node to show you what it actually looks like, because we're going to now talk about T killer cells. And so we need to talk about how does a dendritic cell actually encounter a T killer cell? That's a good question. So if we look at our lymph node, here's one. They're not actually green. That's actually just typical to color them green. I don't know why. The lymphatic system is generally colored green, just to distinguish it from the red and blue that we normally use for arteries and veins. But lymphatic vessels run basically parallel to all of your arteries and veins. They're kind of like if arteries and veins are a highway going either direction, then the lymphatic vessels are a one-way road that actually travels along there, and they end up at these various little junctions that we call lymph nodes. They vary in size, but lymph nodes' job is actually to just sort of be a place to hang out. And that's what we've talked about before, right? That's the cool place to be. Lymph nodes are where macrophages and all of your lymphocytes actually end up hanging out. And if the correct ones have the right combination of things like MHC class 2 and T cell receptors, and those are the big ones here, if they have the right combination, then they can talk to each other and have something cool happen. So I like this thing here because it just shows you the flow of lymph. It is a one-way flow. But your veins and arteries can also connect in here. So you've got these little endothelial venules and the blood capillaries that actually come through these little germinal centers. So this germinal center is the same thing that I showed with B cells last time. And so that's kind of what's happening here, where those B cells develop. And then why would you want that? Well, if you have all of your B cells kind of hanging out in here, and if they trigger an immune response, these germinal centers grow, so they actually swell, which is why your lymph nodes get swollen. And when they swell, they start secreting antibodies, right? And you want your antibodies to head out into your blood because that's how they're going to get everywhere. And so you actually have these vessels here to basically make that happen, and to allow the B cells and the T cells and the macrophages too to have an easy access to the rest of the body.
Structurally speaking, your lymphatic system consists of three main parts. The first is just your lymph. It's a watery fluid that flows through your lymphatic system. Much of your lymph starts out as blood plasma that gets forced out of your capillaries way down in the capillary beds, where the leakage helps the exchange of nutrients and gases between the blood and the capillaries and the hungry cells in your tissues. The blood capillaries reabsorb some of the fluid, but a lot gets left behind. So much of the rest of your lymphatic system works to soak it up and get it back to where it needs to go, inspecting it along the way. For example, you have a network of lymphatic vessels that help reabsorb the fluid at about 600 or 700 lymph nodes, which are basically checkpoints that monitor and cleanse lymph as it filters through. Then there are special lymphoid organs like the spleen, thymus, adenoids, and tonsils, as well as other lymphoid tissues throughout the body. Some of these organs and tissues house special defensive cells of the immune system to fight infection by gobbling up bacteria and other harmful antigens, all of which we'll get a lot more into in upcoming lessons. Others, like the thymus and spleen, provide a nursery for maturing immune cells called lymphocytes. But to understand the special supporting role that your lymphatic system plays in your body, let's start with its origins in the capillary beds of your circulatory system. You'll recall that these beds are where the capillaries carry blood from your arterioles and your arteries before that, and feed blood into your venules and eventually your veins. This is where the transfer of nutrients and waste takes place, as blood pressures force plasma out of the arterial end of the capillaries and into the interstitial fluid between the cells of whatever tissue you happen to be in. And most of that fluid gets drawn right back into the capillaries on the venous end thanks to osmotic pressure. But some is left behind. Of the 20 liters a day that's forced through the arteriolar end of all the capillaries all over your body, only 17 liters is taken back up by the venous end. Those other 3 liters? That becomes your lymph. It's almost immediately picked up by nearby lymphatic capillaries that wind their way in and among the blood capillaries, and they're really well suited for their job. Lymphatic capillaries are made of loosely overlapping endothelial cells that look kind of like roofing shingles, forming little flap-like valves that only open in one direction. So when the pressure in the interstitial space becomes greater than the pressure inside the lymphatic capillary, the flaps push open and take in fluid to relieve the pressure. Once the fluid is inside a lymphatic capillary, we can officially call it lymph. Then it flows through successively larger lymphatic vessels to collecting vessels and then the larger trunks, and finally into one of 2 big ducts that feed back into the lowest pressure area of the circulatory system. The right lymphatic duct drains all the lymph collected from the upper right area of the torso, as well as the right arm and the right sides of the head and thorax, and feeds them all into the internal jugular vein. The larger thoracic duct takes lymph from the rest of the body and dumps it into the subclavian vein. Overall, these vessels work a lot like your veins do. They operate under very low pressure, have a series of valves that prevent backflow, and are helped along a bit by smooth muscle in the vessel walls that keep the fluid moving. Okay, so the lymphatic system takes extra fluid that's squeezed out of the blood capillaries and basically mops it up. That sounds handy, but as a living thing that wants to stay alive, what's in it for you exactly? Well, remember, we're talking about recovering 3 liters of blood fluid per day. That's more than half your total blood volume. If that weren't returned to your circulatory system, you'd be looking at a serious drop in blood pressure and little to no oxygen delivery or waste pickup. Plus, any blockage in the lymph system, like from a tumor that obstructs one of your vessels, can be really dangerous, resulting in edema or swelling that can constrain blood flow and lead to serious complications. And finally, don't forget that your lymph also plays a key role in the function of your immune system. Say you scrape your elbow and some bad stuff, like the Staphylococcus bacteria that's covering your entire body right now, infiltrates some of the loose areolar connective tissue under your skin. Since that tissue is full of interstitial fluid that drains into your lymphatics, which ultimately drain to your heart, you're gonna want to vanquish those bad guys before they have a chance to wreak any havoc. Whatever will you do? Well, this is where your lymph nodes come in. Your body's main lymphoid organs, the lymph nodes, are where your lymph gets close and regular inspection by its resident immune cells, lymphocytes. Depending on what kind of troublemaker they find in the fluid, the lymphocytes might trigger the release of resident macrophages to attack unwanted microorganisms, or they might activate the general immune system outside of the node if they need extra support. The details of how that happens we will get to next time, but for now, just understand that lymphocytes are found in and mature in the loose reticular connective tissue that makes up a large part of the nodes and most of your other lymphoid organs. Now, if a node is overwhelmed by whatever invader it's trying to quell, it may become inflamed. You can actually feel some of your nodes if you poke around the sides of your neck. They feel like little bumps. Little bumps. Yeah, there they are. And noticeably enlarged lymph nodes are often an early diagnostic sign of disease or infection, which is why doctors are always feeling around under your jawline during checkups. But your lymph nodes aren't the system's only checkpoints. You've also got a few special outposts placed at strategic entry points to particularly sensitive tissues, like those in the respiratory and digestive tracts. These are called mucosa-associated lymphoid tissues, or MALTs, because they can be found in mucous membranes around the body outside of the lymphatic vessels. Your tonsils are some of the largest collections of these tissues, which together form a ring around the entrance of the pharynx. There, they can inspect everything you eat and breathe and try to remove any pathogens before they enter the GI tract or lungs. It's a pretty big job, actually, which is why sore throats and swollen tonsils are a common sign of a cold or other infection. Another collection of MALTs, called Peyer's patches, are in the distal portion of the small intestine, providing another checkpoint along the GI tract. And contrary to popular myth, your appendix is not useless because it too contains a bunch of lymphoid tissue and is conveniently located where it can destroy any remaining bacteria before it can breach the intestinal wall during absorption.
So the next thing we actually need to talk about are T killer cells. And so to actually talk about them, we're going to back up a little bit because there's a subtlety that I didn't talk about before. So if we look at this chart, we can see that we normally have, let's say we want to make a B cell response. I reiterate this all the time because it's so important, but we have our macrophage or our dendritic cell that talks to a T helper cell and activates it. And that T helper cell then goes looking for that exact same MHC with that exact same antigen bound into it or held by that MHC class 2 using its T cell receptor, and it can trigger that immune response. Okay, awesome. What I didn't talk about is what actually happens the other direction. Okay, when a dendritic cell activates a T helper cell, that T helper cell also sends another signal back to that dendritic cell. This is why T helper cells are called helper cells and why they are the cornerstone of the immune system. They do something that we call licensing. And so it's a way to, it's kind of like another way of saying activation, but those are kind of different processes. But basically you want your dendritic cell to be able to trigger a T killer cell response. But that's a big deal. To be able to tell a T killer cell to go and blow up cells inside the rest of the body is really dangerous and powerful. So I don't want my dendritic cell talking to any random CD8, okay? I don't want them triggering an immune response, because what if this T killer cell is actually going after self-antigen, just like a B cell could? Well, that would be really bad, right? And so we send our T helper cells through our training process to make sure that their T cell receptor is really specific for MHC class 2 with that antigen. Well, the dendritic cell can then become licensed if it matches. Okay, so we have an awesome signal occurring here. A licensed dendritic cell is now licensed to go and prime T killer cells. And basically you're turning them on and telling them to, hey, go do your job. I realize there's a lot going on, and this is why immunology is kind of a complex process. But if you just kind of sit here and think about it, okay, why am I doing all this? Well, it's these checks and balances because the immune system is incredibly powerful and the lymphocytes are immensely powerful. I do not want them attacking self-antigen, okay? That's why my T helper cells are so stingy about what they activate. They have to have this incredibly specific T cell receptor that can only bind antigen that has MHC class 2, and in this case, activate a B cell. And we also train our T cells to only be able to follow through that. And they have to be able to be turned on or activated or primed, whatever term you want to use, by a licensed dendritic cell. So there's another layer of protection there. There's a lot that I'm not talking about, especially how these cells are interacting, not just with their T cell receptor and their MHC, but with some of these other bindings that can happen. Most notably would be CD28, which is on T helper cells, and then CD80 or CD86 on whatever else it's binding to. And so your T helper cell would normally bind to with its T cell receptor, would normally bind to MHC class 2 with the antigen that it recognizes. And then it also gets a secondary signal via CD80 onto its CD28. And the same thing happens over here with this B cell with CD80 and CD28 here. And so that's that crosstalk that's happening. And so you kind of have this connection between all of these. T killer cells also have similar bindings. They're just different things. So CD just stands for cluster of differentiation. It's just a surface receptor that's triggering some kind of signal. And those are just checks and balances to make sure that this isn't randomly happening. We need to make sure that my dendritic cell is completely operational and my T killer cell is also completely operational, that it's not cancerous or doing something weird. And so if it didn't have one of these promoting receptors, then it just wouldn't get activated. The idea of having these multiple signals being received by multiple receptors is actually a specific thing that we talk about. It's called the three-signal hypothesis. So the three-signal hypothesis, it's these three signals. So we've got our T cell receptor binding to MHC, okay, in the presence of some kind of antigen. Okay, that's signal number one. And then over here, we're going to have our second signal. Okay, so that would be CD28 with CD80 or CD86. Which obviously there's a lot going on there, but basically you have a second signal. And when I was actually being taught, we called it the two signal hypothesis. But that was glossing over this giant fact that you have to have a set of cytokines, which the next lecture we're actually going to talk about cytokines and extracellular signaling that occurs in the immune system. But those cytokines that actually get secreted are what we call our third signal. So we need to talk about this third signal. And so this third signal is actually what's going to decide what kind of T helper cell is it going to become. And so I'm going to cut to this chart. So there's a lot going on here. I realize that. But what I want to kind of distinguish here, look over here. So we've got our T helper 1 cells, or we can have T helper 2 cells. So they've both received the initial two signals. Okay, so they had an MHC binding and they had a CD28 binding. But we need to decide, does your T helper cell want to sort of trigger like a B cell response, like an antibacterial response, which would be sort of a T helper 2 response? Or do I want to trigger more of a viral response, so like a T killer cell response? And that's going to be more of a T helper 1 response. It's obviously much more complicated than that, but to gloss over, that's just the general idea. And the third signal, those cytokines, that's right here. So if you look, I've got my dendritic cell up here. That's going to secrete a specific set of cytokines that triggers a specific set of transcription factors that tells my T helper cell, hey, you need to secrete A, B, C, D, and E. Or if I want to trigger a different kind of response, I want to trigger transcription factors F, G, I, J, and K. Or maybe it's a combination of the two because I need to decide to have it do different things. But in general, at least early on, you generally learn that T helper 1 versus T helper 2 response, and it depends on that third signal. The last thing I want to talk about are T killer cells. We've spent a ton of time talking about B cells and then making antibodies. I've just spent a bunch of time talking about T helper cells and the kind of different responses they can trigger. But we need to talk about T killer cells. So T killer cells, let's back up to our original diagram. So we've got our T killer cell here. And we need to talk about exactly how they work and what they're doing and why in the world are they going to go around, and why are they called killer cells or cytotoxic T cells? So cyto meaning cell, toxic meaning, I don't know, killing or death or whatever you want to say, T cell. It's going to seek out and destroy infected cells. So we need to talk about what that really means. And so in order to do that, I'm actually going to narrate this video that I found on YouTube. It's from the Center for Advanced Molecular Imaging. And so we see here we have our T killer cell, so we're going to assume that it was primed, it's ready to go, it's going to head out into the bloodstream. And remember, T killer cell's job is to look for foreign antigen expressed on MHC class 1. So MHC class 1 is on every single cell in the entire human body, okay? It acts like a little display, just like MHC class 2 displays antigens. Well, MHC class 1 displays anything inside the cell. And if it's a foreign antigen, this is the last ditch effort of this infected cell to put whatever's killing it on its surface in the hopes that it can save the rest of the body. And so you can see here this T killer cell has recognized that MHC class 2. Let's cut back to that again. So we can see that this T killer cell's T cell receptor has recognized MHC class 1 that has this foreign antigen. And so what's happening is it's triggering the release of cytokines. Okay, awesome. So those are going to cause some inflammation, and they're just going to basically tell other cells like, hey, something terrible is happening. And it releases these two things called granzymes and perforins. Perforins are what we're seeing here. So perforins are what actually form a little hole inside of the cell wall. Okay, so this is forming a hole in one of your cells, but that's okay because the cell has a viral infection. We need to kill it. We send granzymes into the cell, and you can see the cell then undergoes apoptosis. So the last thing I want to talk about was granzymes and what they're actually doing. And it's actually really cool. So what they're doing is up here we have our perforin. So that's right here. So these would be your perforins that are going to form that nice little pore into the cell. And then what's being secreted are different granzymes, and they have different functions. There's granzyme A and M and B and K and all kinds of things. But basically they enter the cell and they go through here. And the key component, if you want to trigger apoptosis, is to head into the mitochondria. So the mitochondria is what controls whether or not a cell undergoes apoptosis through a huge process that we don't have to go into today. But basically, you secrete a bunch of cytochromes and they initiate caspases, and those can then go on to cause the cell to start to break down. And so it sends a lot of interesting things into the nucleus. Now, this is a relatively new chart, and it's still unknown exactly how this process works, which is going to be a common theme in all of biology that you'll encounter. But you could discover that question mark. And so I just want to leave this last thing in your brain is this chart, because next week we're actually going to talk about cytokines, we're going to talk about chemokines, we'll talk about interleukins, we'll talk about other things, interferons, and we're going to talk about all these different secretions that come out of these immune cells and other cells as well. But all these different secretions and all these different proteins, and what do they actually trigger in the rest of the body? How in the world does a cell know where to actually head? That's a good question. How does a cell know to actually turn into an inflammatory cell or a regulatory cell? That's a great question. That's what we're going to talk about next week.